Imagine a future where mining is no longer a destructive force, gouging scars into the Earth's surface. A future where microscopic life forms do the heavy lifting, extracting valuable metals from rock with surgical precision. No explosions, no chemical waste, no tailings ponds. Just the quiet, relentless work of microbes. Specifically, microbial communities embedded in structures called biofilms. For decades, scientists have explored the idea that certain microorganisms, especially acidophilic and chemolithotrophic bacteria, can extract metals like copper, gold, and nickel. The process is called bioleaching, and it relies heavily on a complex relationship between the microbe and its environment. But recent research suggests that what really drives the efficiency of bioleaching isn't just the microbe alone, it's the biofilms, a slimy, protective, and highly organized microbial community that sticks to the surface of minerals like a microscopic mining crew. But what exactly is a biofilm? A biofilm is a structured community of microorganisms encased in a self-produced matrix of extracellular polymeric substances, or EPS. Think of it as a sticky web made of sugars, proteins, and DNA that binds cells together and anchors them to a surface. Like rock, inside this matrix, the microbes communicate, share nutrients, and protect each other from harsh environments. Now, in the context of bioleaching, biofilms act as specialized interfaces between the mineral and the microbial world. The biofilm allows bacteria to maintain proximity to the mineral surface, giving them constant access to the ore they're breaking down. This proximity is crucial. The biochemical reactions that solubilize metals, such as the oxidation of iron or sulfur, are far more efficient when the bacteria are in direct contact with the substrate. Let's zoom in on one of the most well-known bioleaching organisms, Acidophilus ferrooxidans. This bacterium thrives in acidic environments and derives its energy by oxidizing ferrous iron and reduced sulfur compounds. When Acidophilus ferrooxidans forms a biofilm on a mineral surface, it begins to create microenvironments that are even more acidic than the surrounding solution, perfect for leaching metals like copper or uranium. The EPS matrix helps trap metal ions, improving leaching rates and creating a localized chemistry that conventional approaches can't replicate. But biofilms aren't just passive structures, they're intelligent systems. Within these microbial collectives, cells use chemical signals to coordinate their activities. This communication, known as quorum sensing, regulates gene expression across the population, switching on metabolic pathways that enhance bioleaching when the community reaches a critical density. In other words, once enough bacteria have joined the biofilm, they turn into a high-efficiency metal extraction machine. This self-organizing behavior is what makes biofilms so promising for the future of mining. But they are also incredibly complex. Understanding how biofilms form, grow, and interact with mineral surfaces is one of the biggest challenges in microbial biotechnology today. Now, why is this important? Because traditional mining is under pressure. As high-grade ores become scarcer, the industry is turning to low-grade, refractory, and even e-waste materials that are difficult and expensive to process using conventional methods. That's where biofilms shine. They can colonize the most stubborn ore surfaces, penetrate microfractures, and accelerate metal solubilization in ways that no chemical reagent ever could. Take gold, for instance. Conventional gold extraction relies on toxic chemicals like cyanide, but researchers have discovered that certain biofilms, particularly those containing leptospirillum and sulfolobus species, can enhance the biooxidation of arsenopyrite and pyrite, releasing trapped gold particles without using cyanide. This environmentally benign approach is not only safer, but also economically attractive for deposits with high sulfide content. What's more, next-gen bioleaching isn't limited to terrestrial applications. There's growing interest in using biofilm-forming microbes for space mining. On the International Space Station, experiments have already shown that microbes can leach elements like vanadium, gadolinium, and neodymium from basalt under microgravity conditions. And guess what? Biofilms formed in space were more efficient than planktonic cells. That means biofilms could one day help us mine the Moon, Mars, or asteroids. Back on Earth, researchers are pushing the boundaries of synthetic biology to design engineered biofilms, custom-tailored to specific ores and environmental conditions. By manipulating genetic pathways, scientists can enhance EPS production, metal resistance, and leaching enzyme expression. These designer biofilms could outperform natural systems, making bioleaching not only feasible, but scalable for industrial use. Another game-changing application is the use of electroactive biofilms, bacteria that generate electric currents as they leach metals. This opens up possibilities for coupling bioleaching with energy recovery, creating self-powered bioreactors that extract metals while producing electricity. In urban mining, biofilms may soon become essential tools for recovering critical materials from e-waste. Circuit boards, for example, contain copper, gold, silver, palladium, and rare earth elements. Biofilm-based leaching could recover these with higher specificity and less environmental impact than current acid-based methods. But it's not all smooth sailing. There are challenges. Biofilms can be slow to grow, sensitive to environmental conditions, and sometimes difficult to control. Moreover, in industrial settings, scaling up biofilm-driven systems requires advanced reactor designs, optimized nutrient delivery. Yet, progress is being made. Researchers are developing real-time biofilm sensors using fluorescence and electrochemical probes. These sensors allow engineers to track biofilm activity, thickness, and metabolic health inside bioreactors. With machine learning algorithms, we can now predict how biofilms will respond to changes in pH, temperature, or metal concentrations, enabling adaptive control systems that keep leaching processes stable and efficient. And then there's the ecological bonus. Biofilm-based bioleaching can operate at ambient temperatures and pressures, consume less water, and produce less toxic waste. In a world increasingly concerned about climate change and resource depletion, this green chemistry approach may help rewrite the narrative around mining. So, what does the future look like? Picture massive underground bioleaching farms, where engineered biofilms extract metals from tailings and waste rock. Imagine rooftop e-waste collection hubs using modular bioreactors powered by solar panels. Think of autonomous mining units on Martian soil using microbial teams to harvest iron and manganese while recycling oxygen. All of this, and more, could be driven by biofilms. In the end, the story of next-gen bioleaching is not just about microbes. It's about a shift in mindset, a recognition that the smallest organisms on Earth may hold the key to the biggest challenges we face. That instead of tearing the Earth apart, we can let biology do the work, gently, efficiently, and intelligently. Biofilms may be slimy and invisible to the naked eye, but their role in reshaping the mining industry is undeniable. They represent not just a technological advancement, but a philosophical one, an invitation to partner with nature instead of overpowering it. The next time you hold a phone, a battery, or a solar panel, think about the metals inside. One day soon, they might have been harvested, not with fire and force, but with a living film of microbes, quietly at work, deep within the earth,